So this is our last session of the day, and I'll be your chair for this session. Just keep in mind we have everything live streaming as well, so we have an audience out there. And so I'm going to go ahead and introduce our first talk. It is Maddie Heater and Gwen. And they are here from the Gannon COVID Lab. They've done a ton of work over the last couple of years, and so we're excited to hear about their project on genomic surveillance of SARS-CoV-2. Maddie and Gwen?
Hello and good afternoon, ladies and gentlemen. My name is Madison Heater, and I'm the COVID-19 Lab Supervisor and Research Assistant at Gannon University. I'm honored to be here today with Gwen and represent Gannon and our lab director, Dr. Jeanette Schnars, and the COVID-19 lab team. Today we'll be highlighting our recent work that we've done focusing on genomic surveillance of COVID-19 on Gannon's campus and how it's helped to develop the upcoming 2-year project that a recent grant with the Erie County Department of Health will help to fund.
There's no doubt that COVID-19 has managed to change the way we live our everyday lives. Basic policies and procedures within our workplace and within higher institutions across the globe have been altered in an attempt to mitigate the effects of COVID-19. This last school year looked much different from fall 2020 to spring 2021. We were able to lift most of our policies regarding COVID-19 testing and many mitigation strategies. Gannon holds vaccine clinics on campus to provide easy access to COVID-19 vaccinations, boosters, and influenza vaccinations. We still use our LiveSafe app to allow students to report symptoms and submit a request to get tested via our health center. We still utilize our online dashboard to report current isolated COVID-19 cases for Gannon, Erie, and Ruskin students and staff. In addition, the dashboard highlights the Erie and Hillsborough County COVID-19 cases. We are one of the only universities in the area to specially develop an in-house COVID-19 surveillance system where we test our students and staff and have results back within 8 to 12 hours after the specimen has been collected. We followed the emergency use protocol from the CDC, validated our surveillance results with a CLIA-certified reference lab. We utilized qPCR to amplify genetic material. If viral genetic material is present, here.
Our in-house testing has allowed us to organize and publish vital data that looks at COVID-19 prevalence on our college campus. And our first publication, which was published in the International Journal of Environmental Research and Public Health, focused on the 2020 to the 2021 school year, and it specifically looked at COVID in the student-athlete population. Our data revealed that the percent of positive tests between sports were comparable regardless of the risk classification. The student-athlete percent positives were also very similar to the general surveillance percent positive tests. On the contrary, we saw a higher percent positive test for non-athletes associated with the health center test. These individuals were usually ones who reported being symptomatic and or a close contact with someone who was confirmed COVID-19 positive. This last academic year, we saw an increase in non-athlete tests, and athletes were tested much less due to the increase of vaccination rates within teams and a change in the previous NCAA policies and procedures regarding COVID-19. We saw a trend that was continuous in athletes despite this transition in procedures and a decrease in testing numbers overall. On the other hand, we saw noticeable differences between 2021 school year's numbers for non-athletes compared to this last school year. The number of tests nearly doubled. Overall, the health center saw a positive test increase of 2.6%, and general surveillance saw an increase of 3.9% in positive numbers of tests. If I can draw your attention down here, you can see that the percentage of positive tests from the 2021 school year to the 21-22 school year nearly doubled for both non-athlete groups. With a few exceptions, Gannon saw similar trends between number of new COVID-19 cases and percent positive cases for 2021 school year. Generally, the percent positive cases hovered around 14% for the days that we saw peaks in cases. As you can see here, there's a few peaks in cases prior to winter break in both November and December. Positive tests became much rarer following winter break. In contrast, we saw a significant surge in positive COVID-19 tests following winter break for the 21-22 school year, as you can see here, particularly in January. This forced us to delay the start of the semester by an entire week. Because of our surveillance testing, we were able to diffuse a potentially detrimental situation on Gannon University's campus. We did not start to see a drop in positive COVID-19 cases until February. We began seeing peaks in our percent positivity rate in late December into early January. Noteworthy are the peaks within the graph around late December into early February. These results reflect periods of high infectivity and contagiousness within the population, suggesting possible seasonality and emphasizing a transition in variants. Despite an increase in testing numbers due to returning from winter break for the spring 2022 semester, the number of positive cases still reflected an elevated positivity rate within the campus population. Before we transition to talking about variant testing, I wanted to put this particular table on here to show that the mutation panel that we used was utilized to further screen these positive samples for multiple variants. This qPCR approach is able to detect mutations specific to Omicron and Delta among other different variants. This table highlights specific panels we use and the variants that the panels detected for. Here we see a percentage of the amount of each suspected variant between August 2021 and January of 2022. The graph reflects Omicron 21K outbreak within the month of December. We see a decline in the Delta concentration and a shift to Omicron from December to January. The two variants of concern, Delta, which is shown in blue, and Omicron, which is shown in red and green, accounted for a majority of the variants we were able to identify. The unknown variants, which are shown in orange, have to be confirmed via sequencing, but are suspected to be subvariants as a result. This particular graph indicates the frequency of each individual mutation between August 2021 to February of 2022. The Delta mutations, L452R, which are shown in white, and P681R, which are shown in yellow, were common during the fall months of 21, however, faded due to the takeover of Omicron. The H69 and V70 deletion, which are shown in blue, then surged with the Omicron spike. D614G, shown in orange, remained the most common mutation, as it almost always is found in all the COVID-19 variants. This particular figure shows the frequency count of each strain variant throughout the months of August 2021 to February of 2022. Here, a more specific shift is seen between Delta, which is shown in blue, and the surge of Omicron, which is shown again in green and red. Unknown variants are also shown in orange. This is a much higher frequency of Omicron accounted for in our positive test, as seen up here, likely attributed to the increased transmissibility of Omicron as a variant. Omicron 21L appears for the first time in January, and we confirm this via sequencing. Unusual mutation combinations usually require us to use sequencing in order to confirm the type of variant via its lineage. Sequencing is especially important in identifying possible subvariants. Next-generation sequencing technologies, or NGS, are used to essentially confirm what we suspect from our qPCR mutation panels. This phylogenetic tree was produced by NextClade using the analysis of 15 SARS-CoV-2 genomes that we sequenced. Each sample is compared against a reference strain at the root of the tree and other known sequences within the NextClade database. The bolded dots at the edge of the tree represent where our samples lie within 5 distinct lineages of the virus. For example, if you look at the phylogenetic tree where the 20B is located, we see a cluster of Omicron samples that we've sequenced, all right here. This program allows us to visually represent and highlight the strains of the virus found within our community. We're able to screen for and identify new variants and subvariants as they arise. Here, again, we use NextClade program to illustrate amino acid substitutions within the SARS-CoV-2 spike protein. This particular analysis allows us to identify specific shifts in mutational frequencies and compare mutations across variants. We're able to identify emergence of new variants and possible sublineages of COVID-19.
So why is our surveillance so important? Because of our surveillance testing, our student, faculty, and staff population has increased by 995 people last academic year. We've created jobs and have not had to lay off a single employee as a result. Not only are we collecting vital data to present to the Gannon community, but we are also able to communicate with the Erie County Department of Health and rely on necessary information about emerging variants. When we discovered Omicron, first with PCR testing and then confirmed with sequencing on December 18th of 2021, the Erie County Department of Health publicized a media announcement encouraging a shift in public health measures countywide to reduce the spread of the contagious Omicron. Because of all this success in our previous work, we have been able to develop a project funded by the Erie County Department of Health that uses similar methodology. This will allow us to expand our data past March of 2022 into the upcoming 2 years. Our objectives include 1, surveillance via swabs from Gannon's campus and the surrounding Erie communities, 2, additional analysis looking for the presence of influenza A and B, 3, sequential testing to monitor viral load over the course of COVID-19 infection, and lastly, creating a COVID-19 GIS map in coordination with the RSC.
So again, objective 1 is going to be looking at COVID-19 surveillance. So essentially, we'll start with the surveillance portion where we collect nasopharyngeal and oral swabs from Gannon's Health Center and also utilize the Erie County Department of Health and the LECOM mobile unit to surveillance test and take those positive tests for further analysis. When we get those positive tests and we confirm that they're positive, we'll then run our variant mutation panels and take any of those specimens that we're unsure about what variant they may be, and we'll sequence them for further confirmation. Now, this graphic might be a little bit difficult for you guys to read, but I wanted to at least highlight how each step of this project was adopted from our previous methodology and how we can kind of incorporate those ideas of COVID-19 and influenza and what that workflow will look like for us. So again, as you can see, start with our surveillance, move through in processing those samples and confirming positive samples, do the variant screening via qPCR with our mutation panels, and do some library work with that, prep any of those tests that we're unsure about for sequencing, and then get confirmation of those variants. So this is obviously very simplified and it's a very long process, but this at least shows a workflow and what it's going to look like for us.
Many thought that influenza disappeared during this pandemic. However, our initial research showed that not only is influenza still present in the general population, but it is also co-infecting COVID patients. This finding is key to why it is a portion of our newest list of objectives. This objective serves as a continuation of my research that I did last year, as I originally wanted to study the relation between COVID-19 and other communicable diseases. I am planning to continue my education in global health and sustainable development to eventually work in disease prevention in third world nations, so this topic falls perfectly into my interests. We have been fortunate enough to find a protocol that utilizes a very similar process to that of the COVID-19 screening that we just showed, to allow us to test samples for influenza A and B if they were either A, COVID positive to look for co-infection, or B, COVID negative but had influenza-like symptoms. We had run over 800 samples last academic year and found that co-infection is indeed present in our campus population. As you can see, many of our tests were done on COVID positive samples, and we found a co-infection rate of 1.41% through 709 tests performed. When looking at COVID negative but symptomatic samples, our infection rate was much higher at 25.45% across 110 samples. In this data, all positives recorded were positive for influenza A and negative for influenza B. We would like to note that we have yet to find a positive influenza B sample.
So objective 3, again, is looking at the course of infection and how that changes from the initial positive COVID-19 test until the end of their isolation when an individual tests negative for COVID-19. So for example, if someone's symptoms began on 11/1 and they tested positive with Gannon University today on 11/3. They are eligible to test out of isolation on 11/7. This testing from isolation day 6 until isolation day 10 allows us to see the progress of the viral load over the course of infection and how it's changing in each individual. Again, I know that this graphic is hard to see, but I wanted to provide this because this is Gannon's particular isolation and exposure flowchart that's readily available on Gannon's website for students and staff to review, and it just kind of brings you through what to do if you're exposed, if you're symptomatic, if you tested positive, etc. So finally, the last objective, objective 4, is a GIS map that will be developed to highlight spatial and temporal changes in COVID-19 cases and different variants throughout Erie County. And this can be updated as needed as new data is produced. And finally, before I hand it over for any questions, I want to give a special thank you to our previous lab team, Ross Gibson, Mara Bestrom, Austin Hertel, and Dr. Stephen Morrow, who helped to develop this methodology and get this program up and running. And again, a huge thank you to you, Gwen, my current student lab worker. And finally, a huge thank you to you, Jeanette, for helping us as our new lab director to get the necessary funding for all these very important projects. Trust me when I say me and the lab crew have had many late nights that have turned into early mornings, working hard to deliver results and vital research data. Everything we do would be impossible if we didn't work together as a team. And although COVID has changed all of our lives, our team is motivated and dedicated to continue our efforts to keeping the community safe through surveillance and vital data. Thank you so much, and get boosted and get your influenza shot.
Good job, Ryan. Is this yours? Yeah. Okay.
Questions?
Yeah. No, but seriously, get your flu shot if you plan on getting it, because we're seeing it on campus right now, so get it before it's too late.
That was going to be my question. Are you seeing symptomatic people that are testing negative?
It is still going around, so.
I have one more question. So thinking back to last year when you had more samples coming in, were people trying to test out at 5-6 days, and does it seem like they were testing out earlier or later compared to more recently?
So that's a really good question. Depending on the variant and what variant we're kind of seeing in the population, it definitely differs. I know with Omicron, a lot of the time we are seeing, sometimes with specific variants, it takes a day or two after you start to develop symptoms for you to test positive. So on occasion, since your symptoms started earlier, you might be in isolation longer. But even right now, I'm seeing a lot of times with people coming in to get tested for isolation day 6, people's viral load is still extremely high, meaning they're still extremely contagious. So even if you're choosing to release yourself per CDC guidelines, make sure you're wearing your mask when you're leaving on isolation day 6, because you're likely still contagious. So please be careful of that. Great question.
I couldn't quite see from your chart.
Sure.
Do you guys suggest isolation even whether it's COVID or if they're sick with the flu, to still do isolation?
So that's a great question. Typically, I don't know what the nurses say for influenza patients and what their protocol is with that, but in terms of COVID-19, right now Gannon's policy is you test positive either via at-home PCR test, you'll go into isolation, and then on isolation day 6, you have the opportunity to test out if you so choose, as long as your symptoms still are improving. So, and then typically you can test every day leading up to your isolation day 10 to be released from isolation. So.
I have one other question. When you were showing the athletes, why did the wrestling team have so much more testing than everybody else?
So that's a really good question. Probably Austin, honestly.
Oh.
I felt like a lot of the time we were seeing a lot of wrestlers. So for those of you who don't know, Austin Hertel, who was in the lab previously with us, was a wrestler, again a wrestler. So a lot of times when we were seeing the infections go through the wrestling team, I know right before break they had an outbreak in the wrestling team, so that was a lot of the reason why a lot of them got tested. And I just know that every time someone was sick, since they knew Austin was in the lab, they were going to go get tested. Yeah,
Okay.
so
A little bit of bias, a
little bit of bias, but they were smart, so.
So are they going to have that or do they have that for the sports teams this year where they have to get tested before?
No,
NCAA completely got rid of
Got
the
rid of that,
testing.
right?
Yeah.
Yep.
Okay.
So we don't even see a lot of athletes coming in to get tested unless they absolutely have to because they report symptoms. But this was when they were required to test X amount of days per week depending on their risk exposure via sport. So that was the majority of our testing in the beginning of when we got this up and running. So great question.
Any other questions? All right, well, thank you so much, guys.
Thank you. I got more questions.
I know.
Is this yours? No. Okay.
Okay, our next speaker is Scott Zanella from Gannon University about COVID and tobacco.
Hello everyone, as she said, my name is Scott Zanella. I'm a senior biology major at Gannon University, and with my partner Grace Foster, who unfortunately couldn't be here today, we kind of performed this review research on the overlap and effect between recreational tobacco consumption and COVID-19 symptoms under the supervision of Dr. Prasad Dalvi, so it was a great time. So getting into it a little bit, getting into the life cycle, a pretty topical outline of the life cycle of an individual COVID particle. It starts off with viral attachment of its receptor binding domain to the mammalian angiotensin converting enzyme 2 receptor, or the ACE2 receptor, which you've probably heard about in a lot of the research that's coming out. But after that initial viral attachment, endocytosis ensues, bringing the viral particle further into the cell for a more intimate reaction between the inner mechanisms of the cell and the viral particle. At this point, emptying of the viral contents will ensue, so that single-stranded RNA virus, the genetic material, is going to be released into the cytoplasm of the host cell, at which point it will be translated by the host cell's ribosomes into those replication and translation complexes, which do a lot of separate things that I don't want to get into the specifics of. Overall, just kind of irrigating those cellular machinery, taking over that, specifically the endoplasmic reticulum and the Golgi apparatus of the host cell, kind of taking those over to create and package new COVID viral particles, and then exocytose them out the cell, or out of the host cell, in the hopes that those new viral particles will then go on and infect other host cells. So yeah, that genetic material initially that's emptied into the cytoplasm codes for those replication and translation complexes, and then more structural things like the spike glycoprotein, envelope and membrane components, as well as like the viral nucleocapsid and things like that. So this SARS-CoV-2 isn't the first pandemic-causing coronavirus, but it is the most recent, most prevalent right now, with over 80 million cases in the U.S. as of April 2022. So that's a little bit of an older statistic, with close to 1 million deaths in the U.S. alone at that time as well. Symptoms at this point most people are familiar with, things like diarrhea, vomiting, hypercoagulable disease, fatigue, dizziness, dry coughing, breathlessness, and fever. So getting into, that's all about COVID. Getting into a little bit about tobacco. The substance is derived from the tobacco plant within the nightshade family. It was first utilized recreationally, actually in 1st century BC. People began to look into it. They're all listed out there, not all of them, but a lot of them are listed out there. Things like cadmium can be found in it. It's a component of certain batteries. And then polonium-210, which is radioactive in and of itself as well. So yeah, just to name a few. Despite those confirmed carcinogens, a lot of people still take part in this recreationally. Pertaining to a younger population of people, the FDA reports around 1 in 10 middle to high schoolers regularly using tobacco products, which equates to around 2.5 million students nationwide, which is just much more than you would expect. Moving on to the right figure there a little bit is a CDC report pertaining to the different age groups going up all the way from 18 to 65 and above. So just some important things to note. is that the older population has a higher incidence of regular cigarette smoking, with almost like a 0 to negligible amount of people that had reported never smoking cigarettes. So it's still surprisingly high rates, despite the negative consequences that are widely known at this point. As a matter of fact, the CDC cites a tripled risk of premature death in those that regularly use tobacco products, with 480 related deaths per year in the U.S. alone, which is this all equates to around $240 billion lost in medical expenditure, kind of dealing with those subsequences as well. So what kind of piqued our interest in this research was a certain controversy. Some research early on in the pandemic was under scrutiny for some reports that I will get into now. So initially, a dose-dependent increase of that ACE2 receptor was identified in the lung epithelium of smokers, which you can see on the right, versus non-smokers. So you can see the ACE2 receptors there. It's just upregulated in that epithelium compared to the non-smokers. So you would actually expect a greater degree of viral entry into the cells of these smokers as compared to non-smokers. But early research actually showed the opposite. A lot of things were being contradictory, and there was this paradoxical relationship showing that smokers were actually at a decreased chance of contracting COVID. The proposed mechanism for that was a certain nicotinic inhibition of viral entry into the cell, but just not enough research has been done to kind of prove that yet. But so we were looking into this early research, kind of trying to review the information surrounding that paradox and collecting the compounding effects of both COVID-19 and tobacco and nicotine consumption, and trying to restore that perspective on the detriments of tobacco, as a lot of them seem to almost encourage the idea of tobacco use as a protectant against COVID-19 contraction. So just like a quick, very general outline of the things we'll be talking about how both recreational tobacco use and COVID-19 contraction contribute to specific things like oxidative stress, leading to systemic inflammation, and leading to worsened clinical outcomes. And then I'll talk about a few specific things between both the respiratory and the cardiovascular system specifically. We already talked a little bit about the ACE2 expression, but it'll come back up. So getting into some respiratory effects a little bit more specifically. This respiratory inflammation is kind of directly reflected in an effect of COVID-19 at the decreased expression of surfactant encoding genes that can lead to lower moisture within the lungs, a lower amount of tension, actually leading to the constriction of the small airways surrounding the alveoli of the lungs. So you can already kind of see how this is converging on impaired alveolar gas exchange, which is the primary function of the respiratory system, is to provide the body air. So something else that kind of worsens that effect in terms of smoking is the introduction of carbon monoxide. So this toxin can have vasoconstrictive effects as well as that hypoxic effect that impairs gas exchange further. And then COVID-19 has pulmonary fibrosis and hyaline membrane formation, both kind of thickening that alveolar wall, kind of reducing the amount of gas exchange that can be effectively exchanged at that barrier. That is indicated by a loss in endothelial junctions and alveolar basement membrane molecules within the alveoli epithelium, just kind of visually explaining that there. And then you can see how these are contributing to things like COPD, dyspnea and fatigue, and severe acute respiratory distress syndrome, that trademark characteristic of COVID. So all leading to the development of comorbidities or the worsening of COVID. So here's kind of a pictorial depiction of everything that we were talking about a little bit earlier. So up at the top, you see that ciliary degradation and cellular senescence. Vasoconstriction is happening over here with that thickening of the alveolar wall, with that lacking surfactant as well, compared to the healthy side over here. You see these inflammatory cells moving in towards that affected alveoli, all of which are kind of contributing to those worse clinical outcomes. Things like, besides the COPD, emphysema, asthma, and then multiple cancers as well. These are only worsened as thrombin and fibrin are deposited to a greater degree in COVID-19 patients, causing a certain amount of clotting, contributing to that severe acute respiratory distress syndrome. And then the development of these neutrophilic extracellular traps is actually common in COVID patients as well. That can cause disseminated intravascular coagulation present in 75% of COVID patients as opposed to the 0.6% of non-COVID patients. So kind of switching gears into the cardiovascular side of things. A lot of the things that we'll talk about in the cardiovascular effects are kind of reflected in what I've already spoken on in the respiratory system. So first things first is oxidative stress can cause that direct physical damage or injury to the membranes of the heart, as well as causing that inflammation to the system as well. Something more specific to the heart is a modification to the heart's lipid profile. Smoking specifically, the use of tobacco products increases the concentration of low-density lipoproteins within the bloodstream that can lead to hardening and narrowing of the vasculature surrounding the heart, contributing to the development of cardiovascular disease and other comorbidities. This is something else that's directly reflected in COVID-19 patients, as they've been found to have increased concentration of lysophospholipids. These are pro-inflammatory lipids that have been shown to cause anemia, multiple system organ failure, and then an increased amount of disease severity. Vasomotor and endothelial dysfunction is something else that's common among both the effects of tobacco and COVID-19. So smoking decreases the amount of nitric oxide expression, which is normally a vasodilator, so it's going to actually have a vasoconstrictive effect, thereby lowering the amount of blood that can be effectively carried around the body. And tobacco also will increase the amount of ACE2 that's being expressed locally to the heart. That can lead to a further degree of infection at contraction. So just leading to poor clinical outcomes, more inflammation, and a furthering of all of those effects that we've talked about a little bit. And last thing on this slide I wanted to talk about is some electrical irregularities. So nicotine that's inherently brought in with a lot of tobacco products has certain effects on the central nervous system, causing things like increased heart rate, blood pressure, and myocardial work that your heart has to do in order to pump the blood around. So COVID-19 kind of worsens this strain on the heart through a certain stalling in the electrical pathways of the heart that can cause increased phase lengths within the heart's contraction periods, which is pretty cool. So moving on a little bit to a decreased oxygen carrying capacity within the blood itself. Tobacco introduces this carbon monoxide that can actually inhabit the red blood cells, distract them from carrying the oxygen that they need to be carrying, effectively decreasing the amount of oxygen that's being supplied to the body. Other than that, this carbon monoxide has also been related to red blood cell massing, increasing the amount of thromboses I mentioned before, and increasing the blood viscosity, slowing down the whole process. So this effect is kind of reflected right in COVID, as COVID patients have increased amounts of that secreted phospholipase A2 or SPLA2 within their blood, essentially correlating to lower amounts of hemoglobin levels, a lowered hematocrit, and lower than normal levels of oxygen saturation. This all leads to further hypoxic effects, and it can even lead to organ failure in some extreme conditions. So all of those kind of wrapping together can increase the effect or the likelihood of cardiac injury, things like stroke, myocardial infarction, dysarrhythmias, and thromboembolisms, just keeping in mind all of those effects we talked about, like clotting, decreased oxygen carrying capacity, and endothelial dysfunction. So this slide kind of just like wraps up everything that I've been talking about, pertaining specifically to the cardiovascular and respiratory systems, and then effects kind of overlapping on both systems. Just kind of summarizing things up, that tobacco use exacerbates COVID-19 symptoms, things like that compounding systemic inflammation, oxidative damage directly to the cells and the molecules within them, and then ACE2 expression is altered as well. All of that can contribute to compromised functioning within both the cardiovascular system and the respiratory system. So all of this kind of leading to the conclusion that tobacco use isn't an adequate protectant against COVID-19 and shouldn't be considered as such. It's just important to keep that perspective even as we go through pandemic times. So yeah, a big thank you to Gannon and the Regional Science Consortium for giving me the opportunity to come and talk to you guys. But at this point, I can take any questions.
Questions?
Yeah.
research. Any other questions? Okay, thank
Cool,
you very
thank you.
much. State Barron and discussing the effects of cortisol exposure.
All right, hello everyone. My name is Ashley Russell. I'm an assistant professor of biochemistry and molecular biology at Penn State Barron. And I'm going to be sharing some preliminary data from a project that is happening in my lab right now. So, an outline of my talk, I'm going to go through some background information, talk about the aims of some very preliminary results, and then some future directions for the project. So, during pregnancy, the placenta is made. So this is a transient organ. It plays a very active role in pregnancy maintenance. So one of its first major jobs is to modulate the immune system to make sure that the mother's immune system doesn't attack the developing baby, which could lead to spontaneous abortion or a miscarriage. And once it has been established, it provides the main source of communication between the maternal bloodstream and the fetal bloodstream. So it allows for transfer of oxygen, nutrients, and waste between the mother and the developing baby. And one of the key nutrients or elements that are transferred by the placenta is iron. So iron is a key nutrient or key element that is super important, especially for brain development in the growing baby. Gestational iron deficiency leads to increased risk of preterm birth and low birth weight, which are not good. And iron deficiency during pregnancy has also been linked to cognitive impairments in offspring, kind of throughout childhood and adolescence, and some studies even show beyond that. So having low iron during pregnancy is very bad. Chronic stress and post-traumatic stress disorder experienced during pregnancy have also been linked to very similar outcomes, such as the experience of preterm birth, low birth weight, and cognitive impairments in offspring. So we don't really know what the link between stress and iron regulation is, but it seems like there might be some correlation. So there is one study that showed that psychological stress has been linked to low serum iron, but this was not specifically done within pregnancy. So this work is kind of aiming to understand how all of these things kind of link together during pregnancy. So as we know, correlation does not equal causation, right? So we know that there's a link between chronic stress and PTSD, increased cortisol, which is the major stress hormone, and altered iron levels, and chronic stress and PTSD, and changes in iron can lead to increased risk of pregnancy complications and impairments in fetal development. But we don't really know what the mechanism is that kind of brings all of these things together. So one avenue for exploration is extracellular vesicles. So I've been studying extracellular vesicles for like 6 years, and I might be a little biased, but I think that they're like the coolest thing ever. So I'm going to talk to you a little bit about what they are and why you should care about them. So there's three main types of extracellular vesicles. The first are apoptotic bodies. They're very big, and so these guys are formed as a cell is undergoing apoptosis or programmed cell death. The cell starts to break down its nucleus, all of its cytoskeletal elements, and form these big kind of blebs of just full of kind of crap that the cell doesn't need because it's dying. The second type of extracellular vesicle are microvesicles. So these are shed from the plasma membrane, not from dying cells typically, and they just kind of bleb off of the membrane of the cell, and they have some really cool stuff in them, so bioactive nucleic acids and proteins that are reflective of the cell that has given rise to them. So these guys are out in the extracellular environment, sometimes causing havoc, sometimes doing good things, depending on the context by which they're released. And then the third type of extracellular vesicles are exosomes. So these guys are made from a very different biogenesis pathway, but similar to our microvesicles, they contain bioactive messenger RNAs, non-coding RNAs, and proteins. All of these are very small in size. That's kind of an important thing I would like to point out, is that they're typically between 40 and 250 or so, 500 nanometers in diameter, so super, super tiny. And so talking about exosomes and how they are made, we have our cell here. The plasma membrane will bud inwards and form an endosome. And then the membrane of the endosome will also begin to bud inwards and form these things called intraluminal vesicles. And now this structure is referred to as a multivesicular body because there's multiple vesicles living inside of it. And so it can take on one of two different fates. The first is that it can fuse with a lysosome and everything inside of it gets degraded. Alternatively, it can fuse to the plasma membrane of the cell and release those intraluminal vesicles into the extracellular environment as exosomes. So once an exosome or microvesicle has been made and released from the cell, it's really hard to differentiate between the two populations. So the field has generally just kind of lumped them together as extracellular vesicles. And so this is a cartoon diagram of an extracellular vesicle. So you'll see that there's lots of transmembrane proteins here, lots of proteins inside. There's nucleic acids inside. People have identified both RNA and DNA. Recent work has even found mitochondria inside of these guys, which is really interesting. But they are released by every single cell. So every type of cell that has ever been studied so far, bacteria, plants, everything releases extracellular vesicles. And they allow for cell-to-cell communication because they contain these bioactive proteins, nucleic acids, and lipids that can affect recipient cells. So a recipient cell can take up this extracellular vesicle either through phagocytosis or membrane fusion and deliver their contents, those proteins or messenger RNAs, into a recipient cell and have an effect. So our overarching research questions are, does exposure to elevated levels of cortisol alter iron transport protein expression in placental cells? And does cortisol exposure alter extracellular vesicle release and cargo? So we're interested in two main iron transport proteins that are found on the placenta. So if we have our placenta here, we have the maternal circulation over here, and our fetal circulation over here. So on the maternal side, we have the transferrin receptor 1, which is an iron importer that's going to be bringing iron from the maternal circulation into the placenta. And then we have ferroportin 1 over here on the fetal side that's basically taking that iron and delivering it to the fetal circulation. It's an iron exporter. So we're interested to determine if cortisol exposure alters expression of these transport proteins and if it alters extracellular vesicle release from these placental cells, and if these transferrin or ferritin receptors are present in these extracellular vesicles. So our overarching hypothesis is that with exposure to cortisol, we will see decreased expression of our receptors, and we will also see decreased release of extracellular vesicles. So the aims of this study, we have two. The first is to determine the effects of cortisol exposure on iron transport proteins in placental cells. And we hypothesize that these cells will express lower levels of these iron transport proteins. And the second aim is to determine if the iron transport proteins are associated with extracellular vesicles and if their expression is affected by cortisol exposure. And we hypothesize that the target proteins will be present in extracellular vesicles and will be downregulated after exposure to cortisol. So to kind of get at asking these questions, we use a immortalized cell line called BWU cells. They're human trophoblast cells, so trophoblasts are a type of cell that is in the placenta. We expose them to cortisol and first check cell viability to make sure that we're not just decimating our entire cell population, because you can't really conduct experiments with dead cells. Next, we isolate our extracellular vesicles via size exclusion chromatography and characterize them, and then we did some protein identification with Western blotting. So these are our data from our viability assays. Our LDH assay gives us information about the cell death, and CCK8 gives us information about cell viability. So we expose the cells to increasing concentrations of cortisol and find that only at the highest concentration do we see statistically significant cell death, but overall it's really not a whole lot of cell death, right? We're like below 5 essentially, and over here we're really not going too far down with cell viability. So it seems like we're doing something to the cells, but we're not killing them. So we chose that as our concentration to move forward with our further experimentation. So we cultured our cells in a flask and collected the cell culture media up here. The media underwent ultrafiltration, which essentially retains anything larger than a certain molecular cutoff size. Everything smaller than that goes through the filtrate. So all of our extracellular vesicles and extracellular proteins are retained in this portion called the retentate. And then we can put the retentate on a size exclusion chromatography column and separate our sample based on size. So our larger extracellular vesicles, if you think about Plinko from The Price is Right, your Plinko chip just kind of bops around the Plinko board. That's kind of what's happening on a very small molecular level. So our extracellular vesicles are the Plinko chips kind of bonking around, and they come out pretty fast because they're big and bulky and just kind of bounce around. Our extracellular proteins are much smaller, so they are still doing the same thing, but they'll typically move and get stuck for a second. Then they'll get unstuck and move again, then they'll get stuck and then get unstuck again and move again. So they move much slower, so it allows us to separate our samples based on size. So we have our large extracellular vesicles here and our small extracellular proteins over here. So we then characterized the expression of our iron transport proteins intracellularly. And interestingly, this is an N of 1, so don't quote me on this. It is not publishable data just yet. We need to repeat this. But we do see decreased expression of ferroportin here, and it looks like increased expression of transferrin receptor 1 here. So that kind of goes against our hypothesis because we expected both of these transport proteins to be downregulated, but it looks like one is being downregulated and the other may be upregulated. So next we looked at expression of these receptors in our extracellular vesicles. So here we have our ladder, our control vesicles, and then our extracellular proteins, and our cortisol-treated vesicles, and our cortisol-treated protein. So here we see in the control and the cortisol-treated extracellular vesicle fractions, we do have expression of TFR1, and it looks like there is a ton of expression in the protein fraction as well. So it's just kind of being released by the cells and not associated with an extracellular vesicle. And when we look at ferroportin, we don't see that one being expressed in our extracellular vesicles, but it looks like there's just a ton of it out here in the extracellular protein fraction. And then over here, this is just a blot that confirms that we do have successful isolation of our extracellular vesicles. So we see expression of CD9, CD81, and CD63 in our vesicle prep here, and we don't see expression of these markers in our protein fraction. So these markers are indicative of extracellular vesicles that are not present in our protein fraction. So our initial preliminary conclusions suggest that our TFR1 is present in extracellular vesicles, and excessive cortisol exposure upregulates TFR1, downregulates ferroportin, and may alter the way it is expressed in our extracellular vesicles. So our future directions are to repeat all of these experiments so that we can actually really interpret our data, quantitate how many extracellular vesicles are actually being released from these cells, and start looking at whether or not exposure to cortisol induces oxidative stress in these BWO cells, because that has been reported in the literature before, so we would like to confirm that and kind of explore those pathways a little bit further. So with that, I would like to thank my current and past lab members who worked on this project and the funding that we have received to work on this. And with that, I will take any questions.
Coming in from the mother into the placenta, does that kind of augment the concentration gradient, increasing the amount that could go into the baby? Like, do you need the extra stuff at the other side, the thero?
To transport it out?
Yeah, do you need the extra stuff to go out, or does the change in concentration gradient enough to increase the amount that goes to the baby?
That's a good question. So when iron is in the placenta, something that I didn't talk about, it can be sequestered by another protein called DMT1. And so typically you would expect that it's being released by the ferroportin on the other side, but it could be that it remains complex with DMT1 and is released in extracellular vesicles and getting to the fetus instead, but we don't know for sure yet. Yeah, good question.